Hello, everyone. My name is Elan Ness-Cohn. I'm a PhD candidate at Northwestern University in the laboratory of Rosemary Brown. And today I'll be discussing topological strategies for the analysis of rhythmic dynamics in transcriptomic time series data. As the title suggests, this talk will focus on identifying circadian genes in transcriptomic time series experiments. Numerous studies have shown a significant link between circadian rhythms and human health. These findings have sparked an interest in identifying genes under circadian control. As a high-level experimental overview, with the advancement of omic technologies, we can now interrogate the expression of thousands of genes throughout the course of the day, generating giant matrices of gene expressions versus time. Nonetheless, this data presents computational biologists with the questions of how best to extract cycling genes from the sparse and noisy time series data. Not only are we interested in identifying cycling genes, but like any good biologist, we also want to determine which genes' dynamics are affected when introducing perturbations to the system as a means to elucidate biological mechanism, as in the case we see here, where circadian rhythms may phase shift as a result of changes in temperature. Today, I'll talk through two algorithms we've developed to address these challenges: time cycle for cycle detection and time change for identifying differential cycling dynamics between conditions. For background, both algorithms are based off of the same underlying principles as defined by Topological's embedding theorem from the field of dynamical systems theory. Suppose we have a dynamical system, but we don't know the underlying equation. We don't even observe all the variables. E.g., perhaps there's some proteins or metabolites or even behaviors that we just can't measure. All we can observe is some variable x, e.g. the expression of some gene. But we observe this observation x over time for an extended period of time. Topological's prove that if we take the time series of x and perform what is called a time delay embedding on it, where we plot each observation of x at time t relative to its position at t minus tau, at t minus 2 tau, and so on and so forth, that time delay embedding will reproduce up to diffeomorphisms, the original unobserved state space. By up to diffeomorphism, I mean it may stretch, translate, or twist, but it cannot be cut or glued. This is useful for our purposes because no cycle can be destroyed or created. The number of cycles in the reconstruction is the same as the number of cycles in the original state space, and cycles are what we care about in the circadian context. As a quick illustration of how time delay embedding works, we can time delay embed a perfect sine wave, f of t. The state space is defined in two dimensions, x and y, with some lag constant t, in this case 3. As we move along the signal, we see a cycle appear in the state space. Now that we have a sense of Topological's theorem and how time delay embedding works, our next step is to identify genes that are cycling, as well as genes that are differentially cycling between experimental conditions. As the goals of each method are different, the analysis strategies at this point diverge. Time cycle works by quantifying the degree of circularity of the embedded signal, while time change will work to quantify the difference between the embeddings of the same genes in each experimental conditions. For now, I'll focus on time cycle and return to time change. Looking at our now embedded signal, the points clearly look like a circle. However, to the computer, it is merely a series of points. To quantify the circularity of the embedding, we will use persistent homology, an algebraic method for discerning the topological features of data. Essentially, the algorithm works by playing connect the dots. From each point in the embedded signal, disks of radius epsilon are incrementally grown. When a pair of points have intersecting disks, a line is drawn between those points. When a complete cycle is formed amongst a set of points at some epsilon b, we see the initial appearance of a topological hole, a birth event, i.e. a closed cycle lying entirely inside the disks surrounding the embedded data. As disks are increased, the topological hole eventually closes at some radius epsilon d, defining the death event. The persistence score of a topological feature is defined as the difference between the death and birth radii. A consequence of Topological's theorem is that periodic signals will have high persistence, while non-periodic signals will have low persistence, giving a measure by which to test statistical significance against a null distribution. It should be noted that noisy patterns will exhibit short-lived cycles. However, the dominant cycling component is still recovered. As an additional measure to incorporate a pragmatic solution to the detection of circadian rhythms in biological data, a biologically constrained null model was developed for time cycle. A common approach for assessing statistical significance is to compare cycling statistics to a randomized time series. However, these randomized time series jumps from high to low states of expression faster than biological translation and degradation dynamics allow. As a result, these methods may produce unrealistic null models and thus erroneous significance tests. Instead, time cycle's null model is obtained by permuting the finite differences in gene expression between sampled time points. By resampling the finite differences, the distribution of gene expression changes under the null is thus identical to that of the observed data, ensuring that the resulting random time series models biological transcription and degradation constraints. We tested time cycle on synthetic data, as well as 3 distinct publicly available biological datasets, all measuring the same condition. Results on synthetic data demonstrated that time cycle was able to reliably discriminate cycling and non-cycling profiles, as well as was robust to missingness, noise, and non-cycling components in the dynamics. Results on biological data demonstrated that time cycle was able to correctly identify known circadian genes across the datasets, as well as showed high concordance across datasets. I.e., if a gene was identified as cycling or non-cycling in one dataset, with high probability it would be similarly classified as cycling or non-cycling in the other dataset. If you're interested in a more in-depth analysis of the data and results of time cycle on synthetic data, you can check out the work recently published in Bioinformatics. Now let's circle back to time change and how we might utilize time delay embedding to identify differentially cycling genes. Instead of quantifying the circularity of the embedding, as was done by time cycle, time change works by comparing the distribution of the embedded points across 2 experimental conditions, using a statistical test known as the Fasano-Franceschini test, aka a 2D 2-sample KS test. To establish a foundation for the 2D test, let's first quickly review the 1D case. The Kolmogorov-Smirnov test is a non-parametric method for determining whether a sample is consistent with a given probability distribution. The KS test computes the CDF as we sweep along a random variable x in both samples. Kolmogorov and Smirnov's insight was defining the distribution of the quantity d, the maximal absolute difference between the CDF of the set of values, and using this distribution for significance testing. Thus, the test can effectively be used to compare any univariate empirical data distribution to any continuous univariate reference distribution. The 2-sample KS test could further be used to compare any 2 univariate empirical data distributions against each other to determine if they are drawn from the same underlying univariate distribution. Extending the 1D case to 2 or higher dimensions, however, is complicated by the fact that CDFs are not well defined in more than one dimension. In 2D, there are 4 ways, 3 independent, of defining the cumulative distribution, since the direction in which we order the x and y points is arbitrary. More generally, in the k-dimensional space, there are 2 to the k minus 1 independent ways of defining the cumulative distribution function. Peacock, and later the more computationally efficient Fasano-Franceschini test, solve this higher dimensional issue by defining the 2D test statistic as the largest difference between the empirical and theoretical cumulative distribution in the 4 quadrants centered at each observed xy pair of points. Algorithmically, this means that the fraction of points in each sample is computed in each quadrant, and the quadrant with the maximal difference is designated with the current maximum for the specified origin. By iterating over all data points and quadrants, the test statistic d is now defined by the maximal difference of the integrated probabilities between samples in any quadrant for any origin point defined by the samples. Since this 2D CDF is empirically computed, Monte Carlo simulation is used to define the null distribution for significance testing. Overall, the test outputs a p-value with the null hypothesis that the two samples were drawn from the same underlying distribution. In our case, defining the likelihood that the embedded signals show differential dynamics. While time change is still unpublished work, an R package for the Fasano-Franceschini test has been developed and is now available on CRAN. You can check out the package website for tutorials if you're interested in using the test for your own work. Having implemented the Fasano-Franceschini statistical test for the time change algorithm, we went on to test time change on both synthetic and biological data. Preliminary results of using time change on synthetic data demonstrated the ability to reliably discriminate different cycling dynamics across varying noise levels, replicates, and waveform shapes. Further work will look at time change's ability to handle missing data, as this will be necessary from a pragmatic standpoint when working with RNA-seq biological data that may have missing samples at different time points. Preliminary results using time change on biological data looked at differential cycling dynamics in transcriptomic time series data collected by our collaborators in the Alata lab. RNA-seq gene expression was measured every 2 hours in replicate over the course of a circadian cycle in fruit flies. Flies were either held at 25 degrees Celsius in constant darkness or shifted down from 25 degrees to 18 degrees Celsius at the start of the experiment to assess how changes in temperature affected circadian dynamics. Time change readily identified 4 classes of genes: those that are temperature dependent at 18 and at 25 degrees, those that are temperature modulated, and those that are temperature independent. Representative plots of the differential versus non-differential cycling dynamics are shown on the right. Of additional interest was determining how time change may work in a high-resolution, low-noise, many-replicate context. In the biological sense, one could imagine using time change to analyze neuronal group firing dynamics, or for high-throughput fluorescent image analysis. While we didn't have this type of data available, we tested time change on erg rowing machine data to look for potential applications in the field of sports science and medicine. In rowing, biomechanics studies have shown that the dynamics of the force curve generated for each stroke is highly dependent on the timing of the force generated by the legs, trunk, and arms during the stroke. Moreover, the sport itself is very technical in nature, with good technique equating to faster race times. In this case, we see an example from these studies of 4 distinct rowing styles and the accompanying distinct rowing force curves. To test time change, we generated data using 4 different rowing dynamics at 20 strokes per minute, 24 strokes per minute, 26 strokes per minute, and a 26 strokes per minute power 10, meaning strokes with maximal effort for the last 10 strokes of the race. Initial results from time change demonstrated an ability to effectively discriminate differential versus non-differential rowing dynamics. Moreover, the algorithm's runtime is a fraction of a second, allowing for potential real-time feedback to a user. One could thus imagine a scenario in which a remote trainer sets an ideal force curve for an athlete as defined by optimal rowing technique. Time change would then act as a real-time feedback system for the athlete to determine if they were maintaining the rowing dynamics required to improve race times. To summarize, I presented 2 algorithms whose bases lie in the field of topological data analysis and dynamical systems theory. For a given time series, the methods reconstruct the state space using time delay embedding. In the embedded space, Takin's theorem proves that the dynamics of a rhythmic signal will exhibit circular patterns. Time cycle uses persistent homology to identify cycling genes, while time change compares the distribution of embedded points between conditions to identify differentially cycling genes. The Fasano-Franceschini test, the 2D 2-sample KS test utilized by time change, has been further implemented as a standalone R package available to all researchers. With both algorithms, we have shown their ability to accurately and reproducibly detect cycling and differentially cycling genes, as well as presented potential future applications for those methods outside the scope of the circadian field for use in the analyses of general cycling processes. To end, I would like to thank my incredible advisor, Dr. Rosemary Brown, collaborators, Dr. Ravi Alata and Dr. Bill Kath, my thesis committee members, Dr. Eric Anderson and Dr. Joe Bass, as well as my funding sources for all their support in bringing these projects to fruition. With that, I'd be happy to take any questions you may have. Thank you.